All right, looks like we are live, and I want to welcome everybody to Standing for Truth. On Standing for Truth, we host debates, interviews, discussions, and more. If you enjoy the content coming out of this ministry, then please hit that subscribe button. Now, tonight, we have an exciting debate between two knowledgeable individuals. This is going to be a ton of fun. The topic for tonight is common descent versus common design. We have a rational empiricist here and NephilimFree. Now, a couple reminders before I bring in the debaters is we've got a lot of interviews and debates coming up in the next month. We've got Dr. Frank Turek, who will be here on the 14th. So make sure you are here for that. It will be a live event. We'll be taking questions from the audience. We've also got Dr. K, who will be here on Monday for an awesome presentation on embryological development, where there will also be a Q&A there. So please bring your questions. We've got also a nice mix of theological debates and science debates as well. So please check the upcoming live stream section for all your reminders. We've also got on the 15th, the big debate, the age of the earth, Dr. Kenny Rhodes and Dr. Kent Hovind
debating this important topic. So that's going to be a lot of fun as well. Lots to look forward to. So anyways, enough for me. You came here to see the debaters and not myself. So let's bring them in. We've got a Rational Empiricist and NephilimFree. Gentlemen, thank you so much for being here tonight.
Oh, my pleasure.
My pleasure.
Awesome. This is exciting. We've got a great chat already, guys. So please make sure you are tagging me throughout the debate with your questions. We are going to be having a Q&A. So that being said, I'm going to go over the format real quick for everybody. So it's going to be a formal debate. We are going to be starting with Andrew. We're going to have 12-minute opening statements, followed by 8-minute uninterrupted rebuttals. Now, the debaters don't have to take the full 8 minutes, but that would be the max amount of time they can use uninterrupted. Then we're going to have a second rebuttal round of 5 minutes. So 5-minute uninterrupted rebuttals. Then we're going to move into an open discussion where we are going to keep this cordial and equally timed as possible, that where the debaters will be asking each other questions pertaining to the topic. That'll be between 35 and 45 minutes. Then we are going to follow that up with a 5-minute closing statement. And then this is when we get you guys in the audience involved. We're going to have a 25-minute audience Q&A. So please make sure you're tagging me at Standing for Truth. That way I don't miss your question. So anyways, gentlemen, why don't we kind of break the ice as we always do and get to know each other a little bit. Gentlemen, why don't we start with NephilimFree. Neph, thank you so much for being here. A little bit about yourself, what's going on over at your channel. How you been?
I'm doing okay. Thank you, Mike. I've been doing Christian creationist apologetics for going on 14 years. And I love to debate. I have live streams on weekends. And I just love discussing everything related to the biblical worldview, creation, evolution, the scriptures, human history, all that sort of thing. And that's what I do.
Awesome. Well, thank you so much, Neph, for that introduction. It's good to have you here. And a Rational Empiricist, thank you as well. This isn't your first time on the channel either. You've been here for a number of discussions, and they are always edifying and cordial. So thanks again. Andrew, a little bit about yourself. How you been? Are you planning on starting a channel and uploading these debates? What's going on?
Yeah, so I go by Rational Empiricist. Otherwise, my real name's Andrew. I, like Neph, like studying these topics, and I have a huge interest in evolution and biology in general. And let's see, I don't know if I'm ever going to start my own channel. I kind of just like to add to conversations already there. But who knows? Maybe I'll start one in the future. Yeah, that's pretty much it. I don't have as much experience as Neph, so not really much to add.
Awesome. Well, this is definitely going to be one to remember. You both are very knowledgeable on this topic, so this is certainly going to be a sophisticated debate. I'm excited for it. And I want to thank Conrad in the chat for your super sticker. I appreciate the support, brother. So that being said, we got the introductions out of the way. We've gone over the format to the audience. Make sure you're tagging me with your questions at Standing for Truth. And that being said, we're going to hand it over to a Rational Empiricist for your 12-minute opening statement. So whenever you're ready, let me know. I can start your timer and the floor is yours.
Yep, got my script here. All right. Can you hear me okay?
Yes. Yeah. I'm good.
All right. So tonight's debate is on the topic of common ancestry versus common design. Which is the best explanation for the data available for living traits? Which explanation is best supported by the data? Independent origins or common descent? There are numerous ways in which this question can be phrased, and yet, no matter how you outline it, the answer is there, plain as day, for all to see, measure, and repeat. The answer is unequivocally yes. Common ancestry is a real phenomenon and an accurate description of reality. It is both an observable fact and a testable, well-substantiated theory. In my opening, I will lay out the reasons why common ancestry is a demonstrable fact, while also revealing how it is and has been tested as a robust scientific model. Common ancestry naturally predicts a high phylogenetic signal for the major clades of life, such as the groupings within the kingdom Animalia. Within phylogenetics, the field of biology that specializes in discovering patterns of descent and exclusivity in life, phylogenetic signal is defined as the statistical dependence of species character states, otherwise known as traits, based on their phylogenetic relationships. This principle of signal can also be described as a way of measuring the congruencies between various tree topologies. In other words, how much does the data support one branching order than the others? Thus, phylogenetic signal is used to quantify the amount of cladistic structure between taxa and their traits within an analysis. It should be noted that some traits can exhibit higher amounts of signal than others. For example, skin color between animals would tend to carry low signal. This is because the dermal colors of various animals can and do change back and forth quite readily, i.e. their states don't carry as much information about the relationships as other traits. If someone were to do a phylogenetic reconstruction based solely on skin color, they would find no significant agreement of one tree topology over another. This low signal trait supports all of them equally well. If we were to look at multiple traits, both high signal and low signal, common ancestry would predict that the high signal should dominate overall. That is, if there is a unique natural tree topology for all life, we should expect to find a highly statistically significant phylogenetic signal for life's major groupings. This is a clear and unique prediction of common ancestry. Indeed, when we look at the embryological, developmental, morphological, and molecular data, we uncover a staggering picture. Each phylogenetic analysis published in the literature has a corresponding measure of signal attached to it. Over many, many decades, the fact of common ancestry has emerged. The phylogenetic signal of life's major groupings is astonishingly high, too high to be the result of random chance. If random chance cannot explain the hierarchical patterns within life, does separate ancestry do any better? One must first consider the predictions of separate ancestry before testing it, if indeed we are to treat it as a testable alternative to common ancestry. If common ancestry predicts a high signal for all life, separate ancestry, by default, must predict a low signal, even if the major groups of life have unique, independent origins from a supernatural creator. In cars and other designed objects, we find, for example, we see this pattern. Traits are incongruent with each other, left and right, and we therefore see statistically low signal for these items that are obviously not related. Cars and designed objects only resemble each other superficially, what may be termed overall or phonetic similarity, rather than nested or phylogenetic similarity. A person doing a phylogenetic analysis on cars will come up with different tree topologies, starting with certain traits, manufacturer, then color, then engine, then wiring, etc., than someone starting from a different set of traits, wiring, then manufacturer, then engine, then color, etc. If separate ancestry were true, we would expect to see the same pattern in living things. One person doing a phylogenetic analysis on a certain group of major taxa should come up with a radically different tree topology than another person coming at it from a different starting assumptions and different traits. And yet, this is not what we see in nature. The nested hierarchy of life is objective and reproducible. One always finds conifers and angiosperms with seeds, yet no ferns have them. One always finds flowers and fruit in angiosperms, yet no ferns or conifers have them. The same thing can be repeated with vertebrate animals. No mammals have insect wings, no insects have mammary glands, no birds have differentiated teeth, no reptiles have hair or fur, etc. Common ancestry would expect these patterns to emerge, not so for a supernatural designer under separate ancestry. One might ask if any studies have been done to test separate ancestry. Again, the answer is yes. And in almost every case, separate ancestry has failed miserably. For example, in Statistical Evidence for Common Ancestry, Application to Primates, Baum et al. 2016 stated that, in this article, we explore a diversity of statistical methods using data from the primates. We focus on two alternatives to common ancestry: species separate ancestry, the separate origin of each named species, and family separate ancestry, the separate origin of each family. We overwhelmingly rejected both species and family separate ancestry with infinitesimal p-values. We compare these results with those from 2 companion papers, which also found tremendously strong support for the common ancestry of all primates, and discuss future directions and general philosophical issues that pertain to statistical testing of historical hypotheses such as common ancestry. This study specifically found that separate ancestry consistently had p-values lower than 10 to the negative 80th power, a number so small that is equivalent to the probability of picking one particular atom at random from the entire known universe or worse. Separate ancestry has not done any better in any other studies either. In Statistical Evidence for Common Ancestry, Testing for Signal and Silent Sites, Bontrager et al. 2016 proclaimed that phylogenetic trees generated from amino acid invariant site nucleotide data agree with those generated from protein coding data, and there is far more hierarchical structure in amino acid invariant site data than would be expected under separate ancestry. Other studies, such as Blomberg et al. 2003, Fitz and Purvis 2010, and White et al. 2013, have all thoroughly rejected separate ancestry in favor of common ancestry for various groups that creationists would largely consider unrelated, such as primates. Now, my interlocutor will likely point out the numerous quote-unquote phylogenetic incongruencies in the literature and claim victory. Indeed, it is true that, like any scientific theory, common ancestry is not perfect. 3 overall problems facing modern phylogenetics include long branch attraction due to processes such as mutation saturation, which tend to erase signal the farther back in time you look, incomplete lineage sorting, usually the result of short speciation times and random introgressions between different populations, and lateral gene transfer, typically found in prokaryotic species such as bacteria. However, amongst the overwhelming evidence elsewhere, these incongruent traits and branches pose little problem for common ancestry as a whole. First, these incongruencies are typically between lesser taxa, not larger groups that everyone agrees on. For instance, all modern phylogenetic analyses support the grouping of Deuterostomia, containing echinoderms like sea stars and chordates like fish and mammals. Second, these incongruencies are a honing mechanism for places where we need more data to resolve certain relationships. Finding minor incongruencies and claiming they diminish common ancestry as a whole, or even the groupings already robust and well established, is equivalent to saying general relativity does not explain gravity at the level of subatomic particles. Therefore, gravity at the macroscopic level does not exist. Obviously, this sort of logic is nonsense. Gaps always exist in large scientific theories, but this does not mean the entire theory is wrong or unreliable. This is how science works. Finally, amongst the larger number of congruencies, the incongruencies we do find are simply evidence that our current picture of a bifurcating tree of life is more complex than we previously thought. In short, they show us that our current representation of the tree is incomplete, not the tree itself. In conclusion, common ancestry is a robust and extremely well-supported scientific theory. It explains the observations we see and does so so well that it is an observable and demonstrable fact of nature. It predicts a high phylogenetic signal for the major groups of life. We observe this in the phyla of the kingdom Animalia, for instance. Separate ancestry predicts a low phylogenetic signal for these groups, lower than expected by chance. Obviously, we do not see this in nature. Finally, many statistical tests have been done against this data, and they have overwhelmingly rejected separate ancestry in favor of common descent. Separate ancestry is simply not in line with the available evidence. Until these models are robust enough, if ever, to combat the theory of common ancestry, one is plainly superior to the other. Thank you.
Awesome. With 2 minutes to spare. So I appreciate that, Andrew. Clear, concise opening statement. We will put that extra 2 minutes into the discussion portion. So that being said, before we hand it to Neph, we got more people in the chat. So please make sure you are tagging me with your questions at Standing for Truth. So that being said, we're going to hand it over to Neph for his 12-minute opening statement. And I've got your screen shared, Neph, so the floor is yours.
Okay. So evolutionists make all manner of assumptions when it comes to phylogenetics. I'm going to address those. But first of all, I think it's important to note that my opponent says one of his strongest evidences is that no lizard, for example, has hair. And all mammals don't, and no mammal has this feature, and only this kind of creature has that kind of creature. That would fit perfectly with the common designer theme as well, because the creator apparently designed organisms so that they had the features necessary for the lifestyle and the ecological niche in which they were going to fill. So it's not true that the similarities of creatures, according to phylogenetics or evolutionary ancestry and clades and whatnot, only supports evolution. It doesn't. It also supports a creation model, but it supports evolution if you believe evolution is true. So firstly, I want you to understand that life was designed. And since life was designed, evolution and nothing that evolutionists put forth as evidence of evolution can be evidence, actual evidence of evolution. What you see on the screen is a mechanism inside a molecular machine that forces with tiny, tiny levers and latches a molecule to take a finished shape. It's mechanical, it's designed, and it modifies a molecule. You can see the levers and the rods that push. This is mechanical engineering at the molecular scale. And there's no method by natural selection to create such a thing. It's utterly ridiculous to believe natural selection could create what you're seeing on the screen. This is one of the methods of DNA error correction or repair. Numerous molecular machine components come into the DNA molecule in specified order and exact locations to perform truly astonishingly complex functions in order to repair a mutated gene. To believe that natural selection could form these molecular machines and the ordering in which they proceed to repair the DNA is preposterous. This is helicase. It duplicates your DNA molecule. It's got scissors, clamps, rotators. This is design, people. This is machine components. This is not something that arose by natural selection. So one of the proofs that evolution isn't true is that the recombination hot swap locations in our DNA do not align with chimpanzees. If you were in a helicopter 3,000 feet above the city, and you look down and saw all these cars coming and going from specific places and wondered, what are those? You come down lower and you see, oh, those are convenience stores. Lots of cars coming and going from those spots. That's like the recombination hot spots in your genome. There are hundreds and hundreds and hundreds of them. They're places where lots of genetic information is passed on, and none of them align with human beings and chimps, not one. For example, in this article published in Science Daily, in a new study funded by the Wellcome Trust and National Institutes of Health, the researchers found that there was no overlap in the location of recombination hotspots between humans and chimpanzees. This is called the recombination hotspot paradox. It's a paradox because evolution can't be true if it's true. Because if we were related to chimpanzees, the location of our recombination hotspots should have been passed on. For them to be moved around by mutation would be such a destructive event to the organism, it would simply go extinct. So we can't be related to apes. Genome is designed. From this study at the Karolinska Institute in Sweden, discovered that the DNA in our bodies possesses linguistics properties much more complex than the most complex spoken human language. Now, I don't know about you, but I don't believe for a second undirected chemistry is able to create grammar, punctuation, syntax, phonetics, alphabetics, and do this in such a way it's more complex than a spoken human language. From this this paper in Science, we see that DNA is known to be prescriptive information. To be prescribed requires forward thinking. That means the mind was involved in its development. These are just modern discoveries. Evolutionists have to deny a whole plethora of modern discoveries in science in order to hold on to the antiquated idea of evolution. Beneficial mutations are too rare to help evolution. There's no way to create an organism with beneficial mutations. They're too rare. 1 out of every 2 million mutations could be considered beneficial, possibly, and they don't affect morphology. If they don't affect the morphology and anatomy of an organism, they can't make a new organism. If you want to believe that mutations, which are damage events, design these incredibly intricate features of all the living things in this world, you're welcome to do so, but you're not being scientific and you're not being rational. Here's more information about that. Let's see. The waiting time necessary for 2 mutations to affect one gene to make it produce an active product is estimated by a scientist to be 100 million years for human beings. Just 2 mutations, 100 million years. See, this is a problem evolutionists have run into, and they don't know how to handle it because modern science is showing them evolution can't be true. But they're going to say, well, it's got to be true anyway. There's got to be a way to fix this, but there is no way to fix it. How many mutations would there have to be to create a human being from a common ancestor with an ape? Hundreds of thousands, possibly, maybe millions. But it takes 100 million years just to get 2 to work together to produce a good thing. How is that going to work for you? Well, it can't. There's not enough history in the 50,000 times the age of this universe for evolution by mutation to create a human being from the bacteria. Not enough time. So this has been known for a very long time. Haldane's dilemma in the 1930s and 40s, Haldane described this problem. The waiting time is far too long. We are degenerating, according to Alexander Williams, the botanist at the Research Associate, the Western Australia Herbarium. He says, to measure the difference between human and chimpanzee genomes, 125 million nucleotides differerences. If only 1,000 of those have been naturally produced, could produce a human species, that means the other 39,999,000 mutations were deleterious. In other words, we're going downhill. Evolution doesn't build anything. These scientists here demonstrate that gene duplication, a favored mechanism of evolution theory, is a good thing, but it's not. These scientists, I'm not going to read these to you for a matter of time, they admit that mutated genes become detrimental. They're prone to silencing, methylation, a phenomenon known as CG depletion, and non-functionalization. That's what happens to duplicate genes. But evolutionists want you to believe that genes get duplicated and then subsequent mutations modify those to become the new information that causes a new feature to arise. That's just not true. What we observe is they're subject to silencing, methylation, CG depletion, and non-functionalization. That's not evolution. That's the opposite. Allegedly, there's 4.5 to 11 million years between humans and a common ancestor with apes. Anywhere from 4.5 to 11 million years. But 80% of the proteins in humans and chimpanzees are different. That means in that period of time, 80% of our proteins would have had to have been modified by evolution. Here's the problem. In the last recent studies show that 73% of non-protein coding single nucleotide variants and 86% of single nucleotide variants predicted to be deleterious have arisen in the last 5 to 10,000 years. That fits loosely the biblical narrative. So where's the 4.5 to 11 million years? It's not there. It's imaginary time. You see, there is no 4.5 to 11 million years. If human beings got off the ark 4,500 years ago, this data fits it because it means all of the changes known to have taken place to our DNA, to our protein codes, have arisen just in the last 5 to 10,000 years. So our evolution didn't happen. There is no millions of years. You see how evolution contradicts real science? Real science shows us that 5 to 10,000 years is the limit for for the mutations that changed our protein codes. But the evolutionists claim humans evolved from apes 4.5 to 11 million years ago. I mean, we had a common ancestor that long ago with them. See, evolution, this is science over here, and this is evolution. Now, phylogenetics. Linda Walkup wrote, the molecular taxonomists have been drawing up evolutionary histories or phylogenies for nearly every kind of life. They're going to have to undo all of their years of junk DNA-based historical reconstructions and wait for the full implications to emerge before they try again. Carl Woese, the famous scientist, wrote, "No consistent organismal phylogeny has emerged from the many individual protein phylogenies so far produced, and phylogenetic incongruencies can be seen everywhere in the universal tree, from its root to the major branchings within and among the various taxa that make up the primary groupings themselves." And also, this study says, incongruencies across different data sets is not a novel observation within mammalian phylogenetic analysis. Therefore, as a final test we perform, et cetera, what they're pointing to is incongruencies in the evolutionary theory assumptions about how similar our DNA is, which is proof that we're related to a particular creature or closely related to this one, less closely related to that one, have all manner of problems evolutionists don't talk about. They'll talk about phylogenetics as though it establishes evolution as a fact. The problem is they never talk about the thousands of incongruencies that are found within genetics that argue against evolution. Here's one. The genes that produce the digits in the feet of birds and the toes of lizards don't align. Different numbers of genes, different Hox genes. That wouldn't be true, because if evolution were true, evolution should have preserved this and passed it on instead of having to start from scratch.
1 minute.
Okay, so there's so much more I could say. I just don't have time to go through this. But what you're seeing on the screen are different studies in genetics relating to teleost fish, bats, all kinds of creatures, and they demonstrate incongruencies across different data sets. It's not a novel observation. Surprisingly, it's commonplace. This is commonplace. So, does phylogenetics demonstrate that life is designed? No, it demonstrates we have similarities based on design, but the problems with it are never spoken about with evolutionists, and there are numerous examples. That concludes my opening.
Awesome. Just on time. Gentlemen, great job to the both of you on your opening statements. Lots of interesting points and topics to get into and discuss for this debate. So that being said, we are now moving into the 8-minute uninterrupted rebuttal. So we're going to hand it over to you, Rational Empiricist. You have 8 minutes, starting on your first word.
Yeah, so several things that were mentioned that were either not relevant to common ancestry in general, or were about specifically the mechanisms of evolution, which is kind of a separate issue from common ancestry. They're kind of different aspects of the theory, if not different theories altogether. The first thing was this whole common design, function, and environment, which of course is why I asked the question. I'm pretty sure I've asked you this question before, Neff, that why are tuna and sharks why are tuna and humans more structurally similar to each other than tuna are to sharks, despite the fact that tuna and sharks live in the same environment? Creationists can't answer that question because they don't have an answer. The only answer they can give is basically that it was the designer just did it that way. And to me, that's not satisfying. That's not a testable alternative. The second thing, again, goes into natural selection evolutionary mechanisms, and how the structures we see in life are extremely complex. Therefore, common ancestry must be false. Well, if you take the evolutionary mechanisms and look at them, sure, they might seem complex, but again, there are different ideas for how these structures can evolve. But again, that's besides the point, because we're talking here about common ancestry, not how did they evolve. We're talking about the patterns overall of, are these things related, yes or no? And of course, they have to be related based on those patterns, not based on evolutionary mechanisms like natural selection or genetic drift. That's not what it's based on. So another thing was mutation models and how there are different types of mutations, beneficial and deleterious, for example. Well, mutation models are accounted for in phylogenetics. In fact, there's different criterion like likelihood and Bayesian inference that are entirely based around different rates of different substitution rates, basically, and different models of how mutations occur and when they occur. So whether they are beneficial or not doesn't really apply within phylogenetic methods. The waiting time problem, here would be my question, for example. Okay, chimps and humans, there's a waiting time problem between them. There are too many differences and too little time. Would the same thing apply to tigers and lions and leopards and other cats, which I'm presuming Nephi would consider closely related? But obviously, there can't be a waiting time problem there. Otherwise, there would be no structural similarities between them. So that would be my question on the waiting time problem. How far back does it go? Generally curious about that. Gene duplication, neo and subfunctionalization were never mentioned, which we know those things occur, and pseudo-functionalization or non-functionalization isn't the only way, the only inevitability of what happens to duplicate genes. So I don't know why you didn't mention those. Let's see. Proteins being different in different organisms does not mean they are 100% different, 80% different, or even 50% different. It could just mean that there are differences between them, which is like, sure, you consider 1% mutation difference between them to be different, but that doesn't necessarily affect the function, because proteins are quite resilient to change within certain limits. So I don't quite understand the quote you mentioned there. So I would like you to explain that if you can. Carl Woe's lateral gene transfer is a big problem, I will admit, at the root of the universal phylogenetic tree. There are a lot of ways to cope with that. But since, of course, we have life, we have no outgroup to life. There's no non-living thing we can compare it to. Otherwise, it wouldn't be living, and we just get into a paradox about that. So since there's no outgroup for life, and the earliest branches near the root of the universal tree frequently undergo lateral gene transfer, exchanging genes between bacteria, archaea, etc., I'm pretty sure Woes was responding to that point, not necessarily the entire tree being falsified. I think he was just responding to the root in general, which is still a problem, but we're getting closer and closer. Like I said, these are honing mechanisms. And the final thing was the difference, I think, which is a crucial difference between what I like to call methodological incongruencies and biological incongruencies. Like, we know there are mechanisms that can create incongruencies. Like I said, incomplete lineage sorting, gene transfer, and saturation and things like that, which we are slowly learning to account for. It's not like we don't know what we're doing here. But a lot of it also has to do with methodological incongruencies and how there's not, like, we've only discovered like 1 million species, or maybe it's a little more, but like, we predict there's probably several million species at least out there. So it's more of a lack of data than a lack of congruence. The amount of congruence already discovered is enough to show that some form of common ancestry is there. I think that's all I want to say.
Awesome. Well, thank you so much there for your 8-minute rebuttal. Andrew with a minute to spare, which we can throw into the discussion portion. So thank you for that. We are now going to hand it over to NephilimFree. Neph, you've got 8 minutes on your first word for your rebuttal. Go ahead. You may be on mute as well, Neph.
Yeah, I'd like to share the screen. Thanks.
Sure, yeah.
Humans more like tuna than sharks. I don't get that. I just don't get that at all. You mentioned, you have this belief, this false belief, this pre-scientific era belief, I would say, that, proteins can accept all kinds of mutations, and this is, they can be changed willy-nilly. No, that's just not true. That's your pseudoscience. Even evolutionist scientists have discovered that just doesn't work. From this paper right here, this scientist states, taken as a whole, recent findings from biochemistry and evolutionary biology indicate that our understanding of protein evolution is incomplete, if not fundamentally flawed. The neutral theory of molecular evolution, which states that all mutations reach fixation in a population are selectively neutral, appeals to evolutionary geneticists in part because it can account for approximately constant rate of protein evolution. However, its premise is that most missense mutations are selectively neutral and has been systematically rejected. Listen to this now. This premise that most missense mutations are selectively neutral has been systematically rejected by protein biochemists, who recognize instead that almost all missense mutations have large biophysical effects indeed. Nucleotide sequencing analysis have uncovered pervasive positive selection for amino acid replacements. Another important challenge to evolutionary theory, which emphasizes the independent and additive effects of mutations, arise from. I could keep reading. So this idea of evolutionists that mutations, proteins can accept mutations kind of willy-nilly and it's all good, is a myth. Because scientists who study proteins and the mutations that occur to protein sequences that produce proteins know otherwise. They know that the vast majority of mutations that occur to proteins cause the protein to fold incorrectly in its shape, even if just a little bit, sometimes dramatically. And when it comes to a protein, because it's so highly, incredibly specific, any change to its conformational shape causes a reduction of efficiency in that protein, or it becomes completely functionless. That's what mutations do to proteins. Proteins don't accept mutations and adapt to them. They get destroyed by them. So it's the myth of evolution versus the science that demonstrates what actually happens to a protein sequence when it takes on a mutation. So common design, since science has demonstrated so thoroughly that life is designed, and I say that boldly and plainly, I can show you examples of that. I've already shown you some. Then we know that evolution isn't true, and the evolutionists, when they see a genetic similarity in one type of life and a similarity in another, they assume the similarity means that they're related. The truth is, it can just as easily mean that those creatures have a certain type of genetic information in them, a certain measure of it, because they need that genetic information for the ecological niche which they were designed to fill by the Creator. So if creation is true and design is true, we expect to find genetic similarities between different kinds of living things. They're going to have similarities in diet. There's going to be a protein similarity. If you eat protein, if you eat something that has potassium in it, you're going to need molecular machines to deal with potassium. Lo and behold, chimpanzees eat potassium. They've got a genetic similarity with humans because we both eat potassium. You see, so we can expect, because of the ecological niche in which we live, because we eat vegetation and we both live on the earth, on the surface of the earth, there's going to be genetic similarities between humans and chimpanzees. See, it's just accepted. That fits perfectly with design. But the problem with phylogenetics is that the more they study it, the more pervasive the problems come. They find incongruencies. I have a quote that I didn't show you, where scientists state that the more they study the genomes of living things, the more incongruencies with evolutionary phylogeny are discovered. It just keeps getting worse, more and more and more. So what we're at is today is this point where the idea of phylogenetics is an old idea, 1960s and 70s and before, right? Evolutionists are hanging on to it still. They're dragging it with them in the future. But the problem is, the more we study genetics, the more problems with the evolution theory, more incongruencies. In fact, for example, we'll find genetic similarity in one kind of creature and another creature that organisms, human beings, the evolutionists believe are common, are closely related. But guess what? The genetic similarity between them doesn't result in morphological difference. Similarity, I mean. Sometimes the genetic similarity in one kind of life and another kind of life, like teleost fish or other things, it doesn't result in a similarity in morphology. But if evolution were true, genetic similarity, the higher the genetic similarity, the more like the morphology and anatomy should be. That's common sense. This is why evolutionists look at human beings and say, look, we have 90 some odd % genetic similarity to chimpanzees. Modern science actually shows it's really more like 86%. But nonetheless, even let's pretend it was 98 or 9%, which it's not. But that's what the evolutionists have been saying for the last 30 years. Let's just pretend it really was. That doesn't relate to the morphology and the anatomy of us. So my example is teleost fish. You got fish with the same clade, and they have fins in different locations of their bodies. But genetic similarity, according to evolutionists, says they're right next to each other evolutionary. They're the same creature genetically, essentially the exact same organism. But look at the difference. See? But they have basically the same genetics, but their anatomy is different. Well, their morphology is different. So genetic similarities don't prove common ancestry. They demonstrate at the very least common design. The plethora of evidence that ruins evolution theory is overwhelming. Today, evolution is a dead thing. This is the beginning of the end of evolution theory. Evolution theory was a naturalist's way of trying to explain the origin and existence of mankind by rejecting the creator. It's a humanist idea, but modern science has ruined it. Because the more we study genetics, for example, the more evidence of incredible design that we find. Information, prescriptive, functional information in the genome, algorithmic processes in the genome spanning across the molecule. An algorithm in the DNA takes information that's located here and is interdependent with other molecular information in the DNA molecule way down there. How does natural selection produce that? Because chemistry works like this. Molecules only interact with each other and could be chemically interdependent with each other only when they're in immediate proximity to each other. So how does genetic information way over here on the DNA molecule become interdependent with information that's way the heck down there? Well, it can't happen by evolution, but it would happen if it were designed. I'll close with that.
Great job to the both of you with 15 seconds to spare, Neph. Great debate so far. Great opening statements and rebuttals. Like I said before, tons of great topics and points. Now we are moving into the 5-minute uninterrupted rebuttal. We've got a ton of great questions flying in, so please keep tagging me at Standing for Truth with your questions. That being said, Andrew, the floor is yours. You've got 5 minutes, and I will start the timer on your first word.
Yep. So the first thing I'd like to say is that, to me at least, design does not equate with common design, if that makes sense. Like, you can have different cars, for instance, made from the same, like they're all designed, but do they have the common designer is the thing. Like, maybe you can tell that they're all designed, but you don't know that they have a common creator, basically. So that's the first thing I would say. Again, similarity is not how we do phylogenetics. Like I said, I made the distinction between phonetic overall similarity, like looking at bats and birds, and they both have wings and they both fly, therefore they're related. That's not how we do things in phylogenetics. We look deeper. We look at the structural similarities, what I like to call nested similarities. It's basically a question of threes rather than twos. That's what I like to say. Basically, you're looking at free organisms or more at a time. finding two that are more similar to each other than to the third one. You can't do that with two, which is why we don't do it with two. We do it with an out group and then in group, and however big it gets. Like, you can have dozens of species, of course, but it runs by threes, not by twos. So it's nested similarity, not just this looks similar to this, we're eyeballing it, therefore therefore they're related. It doesn't work like that. You also have to take into account the matrix you're coding your characters into. For example, you look at multiple traits at a time. Now, of course, some are going to conflict with each other. That's just how reality works. Parts of reality conflict with other parts of reality. Again, incongruencies the incongruencies we find doesn't mean that the congruencies we do find don't exist. I would argue that we actually have a larger number of congruencies, and even if the incongruencies exist, and like I said, they may be systematic or they may be biological in nature, or in origin, it doesn't mean that the congruencies just go away. So there's enough congruencies that we know that some bifurcating pattern exists in nature. Now, some parts of it may be less bifurcating than other parts. That I may say, but basically, when like, you can, I just lost what I was going to say. Third codons, for example. Third codons can vary with, like, they obviously don't have any effect on function of the protein because they don't affect the amino acid produced or encoded for. So under common design, then, why is it that we always find that third codon base pairs show this hierarchical structure, just like in functional parts of the genome? Like, if you're going to say common designs based on function, sure, but if something that's not based on function, the third position of the codon, still varies hierarchically, why is that the case under common design? That's something I'd like to go into, if we can, in the open discussion. And so, yeah, I think I would just say, basically, the things mentioned here, again, have to do with the evolutionary mechanisms that would have produced these traits. But that's not what we're talking about here. We're talking about, is there some kind of pattern that we wouldn't expect under common design? Yeah. I guess the other thing I was going to mention was there are large similarities right now, large differences between chimps and humans, for example. Well, yeah, that can be due to regulatory portions of the genome, which are less studied than other portions of the genome, but we are figuring it out more and more. Like I said, it's all a bunch of honing mechanisms. None of it has to do with what we have already found. I'll leave it at that.
Perfect timing again. Thank you so much for that 5-minute rebuttal. Also, thanks to the chat for such awesome support. Thank you so much for the super chat, super stickers. You guys are the life and blood of this channel. And some great questions coming in as well. So that being said, we're going to hand it over to Neph. Neph, you now have a 5-minute rebuttal, and then we are going to be moving into the discussion or cross-examination round. So, Neph, on your first word, I'll start the timer.
Okay.
5 minutes.
Share my screen, please, sir. Great. So patternicity is what evolutionists refer to, resort to. But again, the incongruencies, orphan genes, the phylogenetic incongruencies, evolutionists don't talk about them. For example, this study, the researchers found that Hox genes that produce the fins in many fish, which are supposedly relatively closely related, don't align, which contradicts evolution theory relating to teleost fish, much less types of fish and reptiles which are believed to be more distantly related. To deal with this problem, the researchers assumed that the genes that developed these features came and went many times in different fish, supposedly explaining the end result, which is that the Hox genes which produce these features are now different genes, though they were once the same. This way, the authors fabricate evidence out of thin air by claiming, oh, they were once the same, and that's evidence that they're still related, even though it's not the same Hox genes. It's not the same Hox genes that produce the fin in this fish that produce the fins in that one. See, that doesn't make sense if evolution are true, especially if those two species of fish are so closely related. Should be the same Hox genes, but lo and behold, it's not. See, so this is the kind of incongruency I'm finding, what I'm talking about. So incongruencies have become pervasive, and it's a big, big problem for evolutionists. They're pretending it's not true. Some might not be aware of it. Okay, but the truth is that the more they study, see, these are more studies. Here's in this particular, in buffalo fish, they find that the morphology doesn't match the genetics of creatures that the fish that they're allegedly closely related to. And here's one about birds and reptiles and the Hox genes that produce the digits. Here's another one between fish and reptiles. And this one, this is a very interesting statement. Incongruencies across different data sets is not a novel observation within mammalia phonetic analysis. Therefore, as a final test, it's not a novelty. They say also, not surprisingly, we found significant incongruencies between the phylogenetic signals. That's the catch term my opponent loves to use. Phylogenetic signals. We found significant incongruence between the phylogenetic signals of the genes and the morphological characters. See, the morphology isn't lining up with the genetic similarity. If you believe this creature is so closely related to that one because the genetic similarity is so great, how come the Hox genes don't produce the same feature in the same location of the body plan in this one as they do that one? See, that's a big incongruency. That shouldn't be the case. If evolution were true, it would not be the case, because you can't just claim that, oh, fix it by saying, oh, well, the Hox genes got moved around, or they'll get deleted and then new ones came. What? The feature disappeared and then regained a new one and the genetic similarity was all fixed? There's no way to fix this problem. See, it's pervasive. The more they study science, the more they find problems for phylogenetics between the evolutionist assumption that evolution is evidenced by the genetic similarity between creatures that they assume are closely related. There's one paper after another. I could show you just a few of these, but there's literally thousands of statements like this in the peer-reviewed papers. Orphan genes, where'd they come from? This creature has dozens of genes that are related, and it's closely related to this other creature. But guess what? That one doesn't have those genes. They're not even in its genome. How's that work if evolution is true? You're saying evolution just deletes dozens of genes and gets dozens of genes more, new ones? How does that work? Evolution is supposed to be progression. It's not supposed to be magical, poof, there's a bunch of genes and poof, you lose a bunch. That's not how evolution's supposed to work. That's not how evolutionists even claim they believe evolution works. It's a progress. It's a process. Mutations occur, slowly modifying genes that change the anatomy of an organism over time. That's what evolution's supposed to do. So where's all these orphan genes come from? Genes that don't have any relative in the creatures that are allegedly, supposedly closely related to it. See, phylogeny is not the end all. It's a problem for evolution, not proof of it. And I'll end with it.
All right, perfect. Just on time as well. So, gentlemen, that concludes the second rebuttal. We have now finished our 12-minute opening statements. We have finished our 8-minute and 5-minute uninterrupted rebuttals. We are now moving into everybody's favorite part of a debate, is the discussion portion. We want to keep this cordial and respectful and as equally timed as possible, asking each other questions pertaining to the points and topics brought up in the opening statements and rebuttals. That being said, Neff just ended with his 5-minute rebuttal. So let's hand it to Andrew. Take a few moments. If there's anything you'd like to address or a question you'd like to ask, go ahead. The floor is yours, gentlemen.
Yeah, so I want to kind of start off with a question I want to ask Neff. So the incongruencies you mentioned, for example, they're kind of at the endpoints. Basically, you have, like I said, incongruencies going further back in time, going to the root of what we would call the universal tree of life. But you also have incongruencies between species. So if, for example, you would accept that some species are more closely related to other, but that ends at some point. So if incongruencies exist between those species, why aren't they not related?
I don't believe in the evolutionists' myth of speciation producing new anatomy. That's what evolutionists believe. So there is no conflict in me. I don't believe that God created one kind of cat, and from one kind of cat came all the different cats. I personally believe that the similarities demonstrate that God is an ingenious designer, that God created more than one kind of cat. I personally think he created small cat species and large ones too. But
Right, but
here's the problem though. There's no anatomical difference between two cats. A house cat and a
wasn't a
tiger
question asked,
have
though.
exactly the same anatomy. Neither one of them has an anatomical feature that's not present in the body plan of the other. They're both fully and anatomically the same. The size difference isn't, or the strength of the jaw muscles, the length of the claws, that's not anatomy. That's morphology. Evolution claims the anatomy changes as time goes on.
Right,
The
but
problem,
that wasn't the question I asked, though, because I wasn't asking, are they related? Did they come from a single species or whatnot? I was asking, okay, you got these incongruencies between these sister species. Now, you would say that incongruencies at larger levels mean that they're not related. So why don't the incongruencies at these levels mean that these sister species aren't related and didn't start independently? Like, that's
well,
kind of the gold standard there.
Okay, I think I answered that question by explaining that I don't believe that all cats are related. I don't necessarily believe that. I don't believe that they're all lizards are necessarily related. I believe God may have created 20 different original kinds of lizard.
But I mean, even between closely related
genetic
species,
similarities
there's still
between them would be
incongruencies.
evidence of design.
Well, I mean, even between like below families, like between genera and between sister species, you can still find these incongruent traits. So why isn't that a problem?
I really don't quite understand your question. I think I pointed out that genetic similarities would be naturally would be expected if God had created creatures, designed
I'm not
them
asking
to
about
share.
genetic similarities, though.
Okay, well, can you reword your question so it's a little?
Sure. Yeah. So I'm saying, so like you have things like incomplete lineage sorting, for example, between certain species, and according to evolution, that would be the result of introgressions and stuff like that. But you would say that that would be those kinds of things at larger levels, say larger kingdoms and phyla, for example, would be problems. Those incongruencies would show that those kingdoms and phyla aren't related to each other. So why doesn't the same thing apply to lower levels between the kinds, between the things within the kinds, which they're still present even there? They're like I said, they're kind of present at the end point of species and present at going back all the way to the root of life. That's where they really predominate, not really in the middle.
Okay,
Why is that?
I think you have a misunderstanding. I don't think that genetic differences between any two types of living thing is an argument against creation. I don't think that's evidence against creation and for evolution.
That
Nor do I
know
if they are closely related or if they're distantly related, according to evolutionists. I don't see a problem with that at all. If we find tremendous or insurmountable genetic incongruency between two creatures that are distantly related, and we find also incongruency between creatures that evolutionists believe are closely related, that doesn't do a thing against intelligent design or creation. That's a problem only for evolution, not for creation. Because
right,
okay.
I'm putting your model up here as a testable alternative. Like, say, take orphan genes, for example. You find orphan genes at the species level. So if they just arise at the species level and they don't have any known precursors, wouldn't that make the species that you would consider related at the end of the flood, or by the end of the flood, wouldn't that mean that they're not necessarily related? It's kind of a double standard.
I'm not sure. You'd have to give me an example, because I don't know of a major incongruency between, or when it comes to orphan genes within a creature that appears to be exactly the same species, like African elephants versus Indian elephants. I don't know of any incongruency when it comes to orphan genes that way. If you have examples, maybe you could tell us about some, but otherwise, I don't know how to address a question that doesn't seem to even relate. It doesn't seem related to.
Well, we can go back to things like incomplete lineage sorting, for example. Like it's you have a population, and then it divides according to speciation, but sometimes if the speciation rate is relatively short, there can be some random side effects going there because genes are just transmitted randomly. So speciation explains that. But however, you would say that incomplete lineage sorting at larger levels, the same kind of incongruencies at larger levels, like incomplete lineage sorting, for example, is a type of incongruency, and doesn't necessarily apply at, say, something as large as a class or phyla, like mammals, for example.
I don't see that as any issue for creation whatsoever. I think I've tried to explain that. But when it comes to something like orphan genes, how would an evolutionist explain orphan genes anyway? Where do they come from? How do dozens of genes pop into existence in a creature? What is the mechanism for the creation of these genes, these novel genes, that are not found in a creature that's allegedly closely related to it, like a particular species of fish that's closely related, according to evolutionists? Where does the dozens of genes come from if they don't have an ancestor, if they're not found in the creature that the evolutionist thinks this species arose from? What's the mechanism for the creation of all these genes? We don't know of any genetic mechanism that can create new functional genes like this. Can you explain to us why we should believe evolutionist science when it comes to orphan genes?
Well, sure, because we have examples of genes coming de novo. For example, you have portions of the genome that are kind of unused and then suddenly become used.
No, that's not de novo genes. That's genetics. That's phylogen. That's simply phenotypic plasticity. The organism already has the genetic information. It's turned on when necessary, and it can be turned off, not expressed. That's an incredible feature of intelligent design. The Italian wall lizards was a beautiful example of that. Evolutionists screened evolution had taken place in only 36 years, but lo and behold, after studying that species in the controlled environment, they found it was only phenotypic plasticity. The organism already had the genes to create enlarged cecal valves and to elongate its muzzle, or to widen the jaw, is not an anatomical feature. That's just a simple adaptation of genetic expression. So that you haven't really given us a mechanism for the origin of orphan genes. Where do they come from? You said that orphan genes can be readily, you alluded that they can be readily produced, but no scientist states that. We don't have any scientific papers that published that show that orphan genes that new genes arise willy-nilly in organisms. It's been observed that, this is a commonplace thing. And it happens, frequently. In fact, a new gene coming into being by de novo that's functional, not that's non-functional or an exact duplicate of another, which is a negative. Duplicating genes by mutation is a negative, not a positive. I can show you sources on that. But you're talking about a new gene that has new function arise. Okay, who observes that this is a commonplace thing? What scientist?
I can't give you the specific name.
Well, there aren't any. No scientist knows of such a thing. By the way, a duplication event that duplicates a gene is rare. It's very rare, and scientists admit that. So even if you had, if this in your imagination, your ideas, a duplicated gene gets subsequent mutations that modify it so it produces new information that does a new thing, or changes the anatomy of the organism in some small way, or something like this, and it becomes functional and preserved. That's not known in science. That's evolution theory. That's philosophy. That's not science. If mutations that duplicate genes are overwhelmingly destructive, and I can show you sources for that, and if they're rare, and they're very rare, I can show you sources for that. Then how in the world does evolution proceed? And how in the world does evolution explain all these orphan genes that seem to pop out of nowhere, right into existence in creatures that are allegedly closely related to each other? Where do those genes come from?
Well, like I said, we see how portions of the genome can arise de novo, and it's not due to
who published
duplication.
That? Who says that? Who shows us that?
Well, I don't know the specific name, but I know that's out there.
Have you got an idea of how frequently this occurs? Because the geneticists I've read their papers from say that duplicate genes are rare. And now I haven't even evoked
some recent
waiting
stuff,
problem.
But I couldn't tell you the exact date. Yeah, I know it's more recent than probably the papers you're reading.
Now, I don't think that you're going to overturn, with this idea that there's some guy has published something that overturns the last 25 years of genetic study into duplicate genes. That because you think there's a paper out there somewhere that does that.
Are you saying these results can't be overturned eventually?
No, because duplicate genes are too rare. I mean, if one
they're not rare in plants, for example. We know they duplicate their genomes all the time.
No, duplicating a genome is that you're talking about. You're talking about reproducing duplicity in the genome. That's not the same thing as copying a new gene and having new genes arise de novo. That's a chromosomal. That's a genetic disorder to have duplicates or triplets or quadruplets of the genome in the cell is a negative, by the way, and that doesn't explain de novo genes. That's just a duplication of the entire genome, copy of it.
Okay, well, we also see duplication of Hox clusters in different lineages.
That's been observed. Okay, so what was the result of that? What was the effect upon the animal?
Nothing. They're just
really nothing
there.
What scientist says nothing? Because the sources I have say duplicating genes and chromosomes is destructive. I have sources for that. Let me show you some, because I don't think you'll believe me unless I show you for this kind of thing. I'm looking for it as we talk. I think you're believing some myths here, but you're not actually demonstrating what you believe. What you believe doesn't seem to match the science.
Well, I mean, like things like gene duplication and things like that isn't really related to common ancestry. The methods we're using don't take into account, okay, there's a gene here, it duplicates. I mean, maybe there's something out there, but there isn't explicitly some model of phylogenetics that talks about, okay, you have natural selection acting on this gene or something like that. It's about what you see in the genome itself. Now I have another question for you pertaining to the topic.
Okay, can I share my screen, Donnie, please? Well, on that subject, though, I want to show you some of the sources for these things. Let's see here. Okay, block duplication and chromosome duplication have been I've got sources. Let's see. Here's the sources. This is just some of them. Okay, there's 15, there's 13, there's source 9, there's 1, 2. There I've got 27 sources for these things. Okay, you can see how I annotated them to produce it. This one is supported by source 2, 3, 4, 5, 6, 7, 8, 9, and 10. It's long been known for decades that random gene duplication mutation in general is degenerative. Contrary to the absurd belief of evolutionists that is designer of new genetic information, etc. Where are the sources for that? 1, 2, 3, 4, 5, 6, 7, 8, and 9.
Now I see Dembski and other creationists there.
Okay.
Can you give me an actual peer-reviewed source?
Well, sure. These I don't think any of the sources that I have here are creationists. Let's go through a couple because I know you have doubts about that. So let's see here. 2. Okay, William Dembski is an intelligent design proponent. This paper here, distribution of fitness affects random etc. That's not a creationist. Professor Goldschmidt, PhD, University of California, evolutionary basis for evolutionary biology, University Yale. He's not a creationist. Dr. Lee Spechner is an intelligent designer. William Botanist is an intelligent designer proponent. Haldane, he was not a creationist. These scientists here in this paper published 2003, they're not creationists. So some of my sources are intelligent design proponents, others are not. So I have a mix of sources here. Yingyan Liu and Dan Morin, they're not creationists. They say gene duplications or aberrations of cell division process are more likely to cause malformation and disease rather than selective advantage.
Now, notice how they said more likely, not always.
Right. Okay, right. More likely. Duplicate genes are usually silenced, and subjected to degenerative mutations. Regulation of supposedly duplicate gene clusters and gene families are irreducibly complex and demands simultaneous development of fully functional multiple genes and switching networks, contrary to Darwinian gradualism. So, you're welcome to believe that gene duplication is a good thing, but it's not. It's negative and it's rare. They're typically subject to silencing, methylation, CG depletion, and then non-functionalization. The scientists you see on the screen here, none of these guys are creationists. Roger Lewin, by example, he's an evolutionary biologist turned science writer. So he doesn't believe in creation, but he admits methylation is commonplace. So gene duplication, you have this pseudoscience philosophy that says mutations are common that produce new genes and they'll be functional and preserved by natural selection. And then there's the science that says they're rare and typically subject to degenerative mutation, not positive.
Can you give me what percentage of them are degenerative?
I don't have an exact percentage, but the scientists use the language typically. Frequently,
scientists use language much differently than the average person.
Well, what does typically mean, or often, or most often? What does that term? What does that language mean? That means the majority of it produces this effect. Not the majority of it produces healthy genes that support the organisms.
Right, but that does mean that there are healthy ones and beneficial ones out there.
So here's where the evolutionist discards all of the science and goes for the jackpot. You pull the handle and think, I'm going to be the lottery winner. It doesn't matter that 90% or something, 80% of mutations that duplicate genes are negative. I believe the lucky one is going to outperform all the negative ones. That's like believing somebody can shoot 22 caliber bullets into a car enough times that one of those bullets magically restores it. Now, you can believe that kind of thing, but that's not scientific. That's philosophy, and it contradicts what's observed, tested, and repeated. That's not science. That's your belief system. Shoot 20,000 22 caliber bullets into a truck, and the magic 20,000th bullet undoes all the damage and gets the car a new bumper. You're welcome to believe that. It's not scientific.
Now we're talking about genetic entropy and things like that, which isn't relevant to the phylogenetic patterns we're talking about. Now, I'd like to ask you this question. It is because
it's orphan genes.
Can I ask my question?
Are negative. That's degenerative. Yes, please ask your question.
Okay, so going to the phylogenetic patterns. Now, I'd like you to give an honest answer to this. Why are, for example, you have convergences and homoplaces which are explained by evolution, Like, for example, why are bat wings more similar in structure to bird wings than either are to insect wings, for example? You can look at this again with other traits, for example, like you can see convergences, which is hard to explain under creation, because when you look down into the homoplasies, the convergent traits, you see that they do have precursor structures.
No, I don't think that's a problem for creation at all. Why are the wings of bats more similar to that of birds than insects? Because that's the way the designer made them.
That's not an answer.
Firstly, they're larger, heavier animals. They don't, an insect like a dragonfly is very lightweight. It doesn't have to have the kind of wings that a bat or a bird does in order to fly. See? So,
well, we also have giant insects in the fossil record, though, which did have large wings.
Right, but its body mass in relation to its ability to use its wings to move it is a very different ratio than with a mammal, with a bird or a bat.
Right, exactly. Right? Why is it a different ratio?
That's the answer to your question.
Why is it a different ratio, though?
Because God is creative and designed different kinds of living things.
You can say the same thing and
how does that support evolution then?
I'm not saying it supports evolution.
All you're doing is assuming that evolution is supported because you believe it's a problem for creation. I don't see a problem for creation, but you sure haven't explained how it's evidence of evolution.
Exactly. There are no problems for evolution for creation. Therefore, it's not testable. It's not a scientific model.
You haven't explained the problem for creation yet, but you haven't explained to us how wings could come into being.
I said there are, like, the creation can account for all phylogenetic patterns, sure, but that makes it untestable, and that makes it pretty much useless.
You just contradicted yourself. But you haven't told us how the wings of convergent evolution, is a plausible idea that evolution is going to design the.
That's not what we're talking about right now.
Well, you brought it up when you brought up dragonfly wing.
We're talking about how do you test
you brought it up when you brought up
how you test evolution.
You brought it up when you brought up that why asked the question, why are the wings of birds different, bats and birds more similar to each other than the wings of flying insects? You brought up the convergent evolution idea.
Okay, so the wings of
I've given my answer. So here's my question for you. How can evolution possibly design the wings of insects and then start from scratch again with this process of designing the legs, the limbs of land-dwelling creatures to morph them into to wings? How does that happen by mutation and natural selection? Can you explain that to us? Because nobody knows such a mechanism.
Again, that's evolutionary mechanisms. We're not talking about mechanisms here. We're talking about, is there common ancestry or common design? The mechanisms aren't relevant to the patterns you see.
No, they're pertinent.
They're different ideas. They're independent of each other.
No, not at all. You're attempting to cut the mechanism out of the discussion because you can't give an account for it. I understand.
I could give an account for it at a later time, but that's not what I came here to debate about.
If evolution is true, the genetic similarity between different creatures is a product of evolution. We need a mechanism for that. Okay.
Why don't we do this then, gentlemen? I'll jump in. Why don't we, Andrew? Why don't you ask one or two more questions related to this specific argument of phylogeny and phylogenetics? Sure. We'll allow Neph to answer them to the best of his ability, and then we'll allow Neph to ask a couple questions. If Neph wants to ask a couple questions related to evolutionary mechanisms, then he can do that at that time. So Andrew, you ask a couple more questions, allow Neph to answer, and then we're going to allow Neph to ask a couple questions. Go ahead.
Sure. Yeah, you can have paternity tests, for example, where you wouldn't necessarily need to know the specific mutations and a specific events going on that led. Oh no,
I'm good. I'm just moderating from backstage for a minute. No worries, Andrew. Go ahead.
Okay, sorry. So in genealogical tests, for example, you don't need to know the specific mutations and the specific events to know that your siblings are more closely related to you than you are to your cousins, for example. you can just tell that by the characters and traits, because we know that's what common ancestry provides. So the same thing applies to larger groups such as phylogenetic taxa. So why can't we apply those same methods based on that same methodology to the larger groups of organisms?
Okay. So reproduction doesn't produce new genes, though. See, so the mechanism is reproduction. Okay, that's what
Right.
produces.
The mechanism is reproduction.
Yes?
That's how we tell.
Okay.
Yes.
Okay, great. So the genetic incongruencies between phylogeny, between different species that are a problem for the evolutionist can't be explained with reproduction. The evolutionist has to have a mechanism for the creation of de novo genes, which get mutated by, I've already explained and shown sources that duplicate mutations that cause duplicate genes is a negative, and they typically are result in they become non-functional, CG depletion, methylation, and they're turned slowly into thymine and other problems. Then they become subsequent to mutations that cause cancer and things like that. So that's a negative. They're rare. So what you're assuming is that any genetic similarity you find is evidence of common ancestry simply because we can point to a man and his son and say, look, there's a tremendous genetic similarity between them because they're related. But what you haven't done is shown that the human being, see, this is a card trick that evolutionists play. It's a shell game. You say, look,
that's a method of by twos, not by
genetic
threes,
similarity between the two. We expect there to be a genetic similarity, great similarity between a man and his son. But now the evolutionist has to show. Wait, I'm answering your question. So, but now the evolutionist wants you to believe that the man and his son are closely related to a non-human. That's the problem for evolutionists. Genetic similarity between humans doesn't prove common ancestry for other creatures. So.
Okay, so if I find, for example, the man and the son are more closely related to each other.
So is this question number 2?
Sure.
Okay.
Now I find the man and his son closely related, or the siblings closely related to each other, that they're connected by their parents, and then to their cousins by their grandparents, and then to their however far it goes, basically. And then you get humans, for example. It's a genealogical process, and in order to stop it, you'd basically have to say, here, heredity stops working. So where does it stop working?
Well, it stops working when evolution. Okay. It stops working when evolutionists can't show a mechanism for the creation of de novo genes that cause anatomical change. That's where it ends. You assume that happens in the distant past over vast periods of time, but you don't have a mechanism for it. It's not science. It's what you believe about the distant past. Pointing to the genetic similarity, this is my answer, pointing to a genetic similarity between a man and his son doesn't give us the reason to believe that human beings are related to apes or lemurs. So you need the mechanism that demonstrates that the differences, the tremendous differences between us genetically can be produced by the reproductive process. You haven't done that. No scientist has done that.
You forgot to mention that those differences are in that nested hierarchy that I mentioned.
You believe the nested hierarchy because you believe that genetics is
a circular. Wait.
What you're engaging in now is circular reasoning. See, I believe that the genetic similarity between this kind of life and that kind of life demonstrates that they're related. How do I know there's a mechanism? Because they have genetic similarity. See, but you haven't demonstrated a mechanism.
No, they're separate. One doesn't say there's a mechanism here. Natural selection caused this gene to propagate. That isn't what's being said here. We're simply grouping organisms by their traits and saying, okay, here's this pattern that shows up. Now, what would best explain this pattern?
I say creation does. You say evolution does.
What specific aspect of creation then would predict a nested hierarchy?
I think I already done that. The nested hierarchy is imaginary, but the similarity.
So we find conifers with flowers.
But the similarity between them, this will be the third time in this discussion that I've already pointed out, is explainable by design. If the organisms are designed to inhabit, let me finish my sentence. Let me finish my sentence. If they were designed to inhabit the same environment, the same type of environment or ecological niche, we should expect in similarities by design. Okay.
Again, you contradicted yourself because now sharks and bony fish aren't similar in structure. Therefore, common design is false because they live in the same environment, but they're not structurally similar.
No, they are structurally similar. Are you saying bony fish and sharks don't have tails? They don't have a mouth. They don't have two eyes. They don't have fins.
Well, I'm saying
there's tremendous
nested hierarchy you're pointing out there.
Because
they
there's
eventually have something similar when you go back far in time, but bony fish have more in common with humans than they do with sharks. Why is that?
Well, because they were designed. But now, if you're going to believe they weren't, you have to show us a mechanism that can create that difference. Nobody's done that, and you can't do it.
No, I'm not trying.
You can believe what you want to, but you need to show a mechanism. You need to show us that this is science, not just what's in your head. It's just in your head.
Common ancestry.
Well, that's not a mechanism.
So you're saying if parents reproduce and pass on their traits, that those traits won't be uniquely inherited by their offspring in subsequent generations.
The problem is you're playing what you're doing now is you're playing that evolutionist card trick again, you see? Because there's genetic similarity, it means that they're closely related. You haven't shown us
I'm not saying genetics just similarity.
Let me finish my statement. You haven't shown us how genetic similarity between creatures that are distantly related to each other is a mechanism to create the that difference or that similarity. You haven't shown that. Pointing to the fact that humans have babies that have the same genetic information in them.
Not the same.
Doesn't demonstrate that humans are related to a lemur. See, if you're going to say we're related to a lemur, you've got to show a mechanism for that.
Right. That example within humans provides a mechanism, and that explains, okay, here's what we see under common ancestry, a miniature version of common ancestry, but here's what we see under the principle of common ancestry. Now, we see the same effect in living in larger groups of life, so common ancestry would explain that.
Now, reproduction, ancestry, that's observable. But your idea that this kind of creature is related to that other kind of creature, or humans are related to lemurs, that's not observable. See, so you're just assuming that that we are. That's circular reasoning of evolutionists. You haven't demonstrated you haven't demonstrated there's a genetic mechanism that's going to move a human being away from being a human or bring a human being into existence. The similarities or the differences between a man and his son are insignificant. Genetically insignificant, but you're talking about great distances of time and great genetic differences to produce. The boundary is designed. The genetic information is designed. If you're going to say that that mutation and natural selection create de novo genes that have produced new functions and change anatomy, so that your belief about what happened in the distant past can be considered science and not philosophy, you need to be able to show examples of that occurring in the real world. I've asked you for that twice now. I don't care about your circular reasoning. You're welcome to believe we're related to some creature that lived, 4.5 million years ago or something. But if you're going to say that genetic similarities between a man and his son has no limitation, and that genetic change will continue to create this tremendous difference between a human and a lemur, you've got to be able to show a mechanism to make it scientific. The mutation creates de novo genes that are transformed into new information that affect the anatomy of a living thing. Your beliefs don't figure in. That's religious. I don't care about your religion of evolution.
But here's the thing. We're not talking about how the mutations are selected or what order they come in or whatnot, what environment they're found in. We're simply looking at, hey, this is the mutation rate. Now, here's a mutation, let's say A, going to this other base pair, let's say T. What's the rate at which that occurs? That's the probability.
Now the mutation rate is. I'm sorry, I interrupted.
Now here's the thing. So like under optimality criteria such as likelihood. You can go in the genome and you can find, okay, here's the probability of this base pair mutating into this one. So if this one's a high probability, that's not really useful because it could happen all the time. But if you find a low probability one and you do find that between two closely related species in comparison to another one that doesn't have that mutation, that suggests that that mutation arose in the common ancestor leading to those two and not in the third one, the outgroup. That's what we're looking at here. We're looking at by threes, not by twos. We're looking at nested similarities.
What we're looking at is your philosophy about what happened in the distant past without a mechanism.
So you're saying
I've asked you for this mechanism already. You haven't been able to provide a source for it. Nobody knows of any such mechanism. And you say mutation rate. The mutation rate, as I've already explained, and I could show you other sources, is way too slow for mutation to create the information necessary to make a fish into a land-dwelling creature. Let me finish. The waiting time problem is too great. I showed you a scientist who is a secular scientist who points out in his paper that the waiting time is to create the differences between humans and chimpanzees is 100 million years. Now, but you believe human beings are related to apes between 4.5 and 11 million years. But they say the minimum to get one new gene that has a function produced by two mutations, one that creates it and one that transforms it into something that's new and functional, to just two mutations, 100 million years. Can you explain to me at that rate how evolution is going to produce the differences between the human genome and the chimpanzee genome in 4.5 to 11 million years, when just to get one genetic difference between us takes two mutations and 100 million years? Please explain the discrepancy between what you believe in your head and what science has shown us.
Okay, so I'm going to jump in. Okay, so we've got Neff. He asked you a question, Andrew. Now, Andrew, you take as much time as you need for a response or rebuttal to that specific topic. Go ahead.
Right. Yeah. So the waiting time problem. Now, I must admit, I'm not as familiar with it in the details as I probably should be. Now, the calculations, as far as I'm aware, are kind of flawed because they assume a kind of linear mutation kind of lockstep pattern, and to me, that if that is correct, if I'm understanding the problem there correctly, that is that's a flaw in the argument, because we know mutations don't happen this one, then this one, then this one, then this one. We know they happen all over the place, and that's where the genetic differences come from. So that would be my response to that. I don't know the specific details behind it, but that's how mutations work.
And go ahead, Neff, your response.
Well, all you've done again is share with us your philosophy. If I could screen share something real quick. So this is not creationists. This is from X chromosome gene duplication lead to retardation by Michael Smith, North American correspondent, MedPage Today, published 2008. DNA deletion and duplication are associated genetic disorders. Rearrangements of chromosomes due to deletion of DNA sequences and duplicated segments, both of which can be encompassed thousands to hundreds of thousands of bases. Why do large rearrangements occur? For one, et cetera, et cetera. And they talk about genetic disease. So when it comes to duplicating a gene or duplicating a chunk of DNA, a block, they call it, or duplicating an entire chromosome, the typical result is disease or deformity, sometimes stillbirth. So what you're believing is there's a mechanism that explains the genetic difference between human beings, and it happens outrageously fast. It would have to happen outrageously fast if evolution was to produce a human being from a common ancestor with a chimpanzee 4.5 to 11 million years ago. But we can't do anything but show that duplication events are negative and destructive to organisms. and that they don't occur so as rapidly as the evolution would have you believe. You have to believe that what happens, it's been scientifically demonstrated to happen, happens dozens of times faster, maybe hundreds of times faster than what science shows us it does. So it's your ideas versus science.
Okay, let me jump in here real quick. So we had, in these last few minutes, we had Neff ask you a question, Andrew. You responded, and Neff gave a rebuttal to that response. Why don't we allow you now to give a brief response, if you'd like to some of the points Neff brought up. And then if you'd like to, you can ask Neff a question. And then we'll get a couple more questions in from each of you. And then we're going to go into some closing statements. So go ahead, Andrew.
Sure. Yeah. So I would like to clarify on this waiting time problem because maybe I'm not understanding it correctly. I'll admit that. So Neff, are you saying that the waiting time problem is according to, like I said, a linear sequence of mutations, or is it more across the genome, or how is this problem being summarized, basically?
Well, it's just relatively simple. To produce the tremendous genetic difference between humans and apes, for example, requires hundreds of thousands of base pairs, right? So the evolutionist has to believe that mutation occurs at a rate that's going to produce hundreds of thousands of base pairs of information in the amount of time since the common ancestor with chimps, right? But the problem is that genetics, studying the genomes of creatures, even humans, shows that mutations which duplicate a gene, which could then be mutated by subsequent mutations to transform it into new information, which becomes new functional information to help promote evolution, happens at a rate that is vastly too slow for evolution theory to be true.
I don't know that. Like I said, I don't know the specific genetic details behind that, but
well, here's an example. Can I give you an example? Sure. Scientists discovered 250 million year old bacteria in a salt dome, in a salt inclusion. He took those bacteria back to the lab and revived them. Okay. Now the problem with that is two problems with that. One, this is evidence of young Earth creation, and secondly, it demonstrates that the genetic information in the bacteria didn't change significantly in 250 million years. Why? If evolution is true, how in the world could it be true that the genome of a bacteria hasn't changed significantly in 250 million years? For example, here's the study. If I could screen share it real quick so you can see exactly what I'm talking about. This study here. Isolation of 250 million year old bacteria in a primary salt crystal. They found these bacteria, they revived them, and guess what the evolutionist scientists did? They claimed he corrupted his samples. He's a bad scientist. Anytime an evolutionist scientist produces evidence that argues against evolution, what do? They can him, throw him under the bus. He's not a bad scientist. He just produced data that doesn't jive with evolution, and so they have to blackball him. But we know the history of that. That's happened many times. So can you explain, if your mechanism happens so willy-nilly, how can the genome of a bacteria 250 million years not change significantly?
Like, did it say change significantly across the whole genome or across a specific region of the genome?
The genome found in the bacteria is not significantly different from the bacteria that is exact same species living on the Earth today. And these are allegedly 250 million years old because of where they were found in the geologic column. So how can 250 million years pass and the genome of a bacteria not change significantly if evolution is true?
Well, what does significantly mean in this case?
Yeah, there goes the games. So not significantly. Okay.
I'm not saying it's not significant. I'm trying to understand what is significant.
I don't know where to go.
In this case. Like how big the genome is.
Mean? Yeah. What does mutation mean? What does significant mean? We could play word games all day long, but
Okay.
it's a
If
simple
I were to
question.
rephrase my question, it would be asking, by significant, how much of the genome are these differences accounting for? Like, what's the actual number? Because if I don't know that, I don't know what significant means. Because
well, let me just ask you this way: should the genome of an organism change significantly in 250 million years if evolution by mutation is true?
again, I don't know what you mean by
You don't know?
significant.
Well, then you don't have an argument, since you don't know even if evolution should significantly change the genome of an organism in 250 million years. You don't have any arguments for anything then. Thank you for just refuting yourself, because you just blew your whole argument to heck.
Okay, why don't we do this, gentlemen, because we have gone between 5 and 10 minutes over on the discussion portion. It's been really engaging, tons of great topics discussed, so I didn't want to end it prematurely. That being said, why don't we, before we get into the concluding statements, why don't you each take a minute or two just to kind of sum up your thoughts in terms of the discussion, just kind of give your final thoughts, and then we'll go into some concluding statements. So since, Andrew, you started off the discussion, why don't we give it to you, take a minute or two, just kind of wrap up your final points, then we'll allow Neph to do the same, then we'll go into some 5-minute concluding statements. Go ahead, Andrew.
Right, yeah, I guess what I would say is that this discussion is mostly focused around the specific mechanisms of what takes place. How do these changes occur? And to me, that's not what phylogenetics focuses on. What phylogenetics does is measuring the changes specifically, not how they occur or how they're selected for, or how they drift out of the population. It doesn't assume a particular evolutionary mechanism. It's just saying, hey, there are these unique traits that are shared by this subset of organisms, but not found in this larger subset of organisms. And if we go farther, then this larger subset of organisms shares these traits, and so on and so forth. That's the nested hierarchy. Basically, let's see what else. This whole thing about gene duplication kind of falls into that, because, like, we know gene duplication isn't like mutations in general are rare, sure, but rare in this context isn't particularly useful, because again, you have many individuals in a population all at once, and so basically the thing about mutations and gene duplication, for example, kind of seems a bit silly to me, because we have examples of, like, the one I know the most about would be the antifreeze proteins in Antarctic ice fish, and that would be the example of a beneficial gene duplication. But yeah, mostly this discussion overall has focused not on the broad phylogenetic patterns, which I think are just as important as looking at, say, the differences between species A and species B, which should or should not be closely related to each other. And so overall, yeah, that's what I think about this discussion. I don't think it has been as specific as, or as broad or specific as it could be. It's kind of been in the middle.
Okay, I appreciate those final points and final thoughts pertaining to the discussion. Neph, take a couple minutes to give your final thoughts, final points in terms of the discussion. Then we're going to go into 5-minute concluding statements. So anything you want to say in your concluding statement, save for that. This is just to finalize the discussion. Neph, go ahead. And you might be on mute, Neph.
Okay, I think I've demonstrated that evolution is philosophy. That it doesn't. It's not scientific. It's a belief about what happened in a distant past, and it doesn't. It's not science. It's philosophy. And when pressed for mechanisms, observable, testable, scientific mechanism that can create evolutionary change, evolutionists are at a complete loss. I could address the other things that he said just now, the antifreeze genes and whatnot, but I'll save that for the Q&A. I think that's pretty much concludes my statement. No mechanism, just philosophy. It's not science.
Okay, I appreciate that. That was a great discussion, gentlemen, very engaging. We've had a great chat and a very lively chat as well, discussing the topic at hand. So that being said, we started with Andrew. So Andrew, you can have the floor for your 5-minute concluding statement. Then we'll hand it to Neph for 5 minutes, and then we're going to get into about 20-25 minutes of questions from the audience. So Rational Empiricist, go ahead.
Yeah, so to me, common ancestry is a topic that can get pretty heated, because obviously we have this whole discussion about, are we related to apes? Are we related to platypuses? Platypuses? Who knows? Basically, the whole thing about common ancestry is that it's observable. Like, there's a miniature version of it in between, like within species, between certain species that we've observed that have speciated, for example, and between populations. And what the methodology behind phylogenetics is looking for those exclusive relationships and making them more and more inclusive, or in some cases less and less inclusive. You're building that nested hierarchy. And I think this is a good topic because, not necessarily you, Neph, but like many creationists don't understand the concept of a nested hierarchy. And I think sometimes it's important to educate, even if you disagree with someone, and to have a good, entertaining discussion, and to learn from each other. And for example, oops, I looked at the chat. Synapomorphies, apomorphies, plesiomorphies. These are terms that are widely used in the phylogenetic literature, and I mean, even lay people don't necessarily understand what they mean. So it's important that we have these discussions and we talk about these subjects. And the only thing, like I said, that I have to quibble about is the fact that we're talking about these patterns and methodologies, and not necessarily, we don't need to talk about the concept of evolution, or rather mechanisms of evolution, to talk about that. Like, they're separate from each other, and that's another thing that I think is often misunderstood. Like I feel like this whole discussion of common ancestry, for example, you can talk about the methodology behind it, you can talk about the philosophy behind it, etc. And to me, common design in relation to common ancestry is not a valid explanation, because in order for an explanation to be an explanation, to do what it needs to do, it has to give us information about the world that we didn't know. It has to predict. It has to define certain aspects of reality, and to clear those up and make them more and more accurate. That's the whole scientific process. And common design, to me, just stands in the way of that, in terms of saying the designer is creative and made these patterns in this particular way because he's creative and he was just decided to do it that way, is not an explanation, because it does not specify the particular aspects of why he did it, how he did it, what way he did it in, not really when he did it, because at this point, creationists would say that the Earth is 6,000 years old. But yeah, I think that's all I really want to say. This whole discussion is talking about the specific mechanisms, when sometimes they really should be separated and they should be focused on their own. And if you want to say common ancestry is false, then you need to show that in its specific domain that's trying to handle, which is these phylogenetic patterns and these exclusive relationships and these nested similarities.
Awesome. Well, thank you for that 5-minute concluding statement. Let's now hand it to Neph. Neph, you also have 5 minutes. Make sure you unmute yourself, and I will start the timer on your first word.
Okay, I'd like to screen share. I have demonstrated that evolution isn't true, and my opponent can't show a mechanism for evolution. So I've demonstrated problems with evolutionists repeatedly with the theory of evolution. That's not working. So let me stop screen sharing. Let me try that again. So the evolutionist believes that there's a mechanism for evolution somewhere, but nobody seems to know what it is. And the evolutionist, all right, let me try to screen share once more, please. And they believe that there's, can I screen share what I'm, am I screen sharing? Is it coming through? Hello? Can I be heard?
Yeah, I can hear you. Are you there,
Oh,
SFT?
Okay. So I don't, do you see a picture of a turtle?
I did just a minute ago, but now I don't see anything.
You should.
Oh, there we go.
Okay. Oh, okay. So
You're good.
I think I've demonstrated evolution isn't true, and evolutionism is philosophy. What we know about living things is they may change in size, but they don't change in design. The fossil record is rife with examples. That's how big a turtle was prior to the flood. What you see on the right is a modern wombat. On the left is a model, a reconstruction of a pre-flood wombat. Look how big they're anatomically the same. One is gigantic, and the other one is much smaller. Here's a dog. That's how big dogs were before the flood. This is on Discovery magazine's front cover. That's a dog. It's anatomically identical to every dog that's ever lived on the planet. It doesn't change. Evolutionists believe that mutations, which are negative, they deny science. They believe that mutations are a positive thing and they transform things, create new genetic information that transforms creatures, gives them new features and all that. We don't know of any such thing. All we know is that mutation is a killer. That's what it is. It destroys. Dogs prior to the flood weighed 1,000 pounds, at least 600-700 pounds. Now they don't come. They're not anywhere that big. There's a turtle. There's examples. This is one of the largest rhinoceros-like creatures that ever lived on the earth. You see how big it is in relation to a tree? This is an illustration that was in the news recently. They found that's how big some of the creatures in the world were in the past. So a modern wombat and a fossil wombat. The fossil wombat's the same anatomically. He's just gigantic. So creatures don't change. The fossil record demonstrates that. Numerous paleontologists, geneticists, etc., have all admitted, including Stephen Jay Gould, Niles Eldredge, Kemp. The list goes on and on and on. We don't know of a mechanism for evolution. Evolutionists assume that genetic similarities between creatures that are distant related to each other, allegedly, is evidence that they're related. But as I pointed out, that can be explained easily by design. But the evolutionist has to demonstrate that the differences between us genetically can be accounted for by a mechanism that's scientific and not a belief system. But my opponent hasn't been able to do that. None of the people I've ever debated have ever been able to do it. We don't know of a mutation of a mechanism that duplicates genes and creates new genetic information that's functional, useful, and changes anatomy. There is no such thing known in science. So it's an assumption that when we find genetic similarities, it means they're related. But that's easily explained by intelligent design. There is no mechanism for evolution. Evolutionists have no science on their side. They can't show that a genetic similarity between a human being and a chimpanzee or a lemur or any other creature is evidence that we're related to them, as opposed to designed to share similarities in our environment. See, so the evolutionist is welcome to believe that demonstrates common ancestry, but unless you can show a mechanism that demonstrates that we have a common ancestry, the evolutionist has nothing but their philosophical constructs about what happens instead of science, which demonstrates what we can observe happens. Now, I'll conclude with that.
All right. Well, thank you for that concluding statement, Neff. Thanks to the both of you. This was an awesome debate. Definitely one to remember. I had a lot of fun. The chat had a ton of fun. Very engaging discussion. So great job to the both of you. Why don't we get into some questions now from the audience?
Sure.
And if anybody has any, there we go. If anybody has any questions that maybe just came to mind as well, feel free to put them into the chat, and therefore I can kind of put them up on screen. Most of these questions I have were from an hour and a half ago or so, from when the debate was just in its early round. So that being said, let's get to the first question from, let's see here, Frosty Soda. This question is for ARE, so I assume that's you, a rational.
Yeah.
So the question is, are our ancestors fish? If yes, what separates us from the fish that we eat on our plate?
The problem, I guess, I don't know if this answers your question, but basically, fish is technically paraphyletic in that, in a sense, you can say, okay, according to evolution, all these things that we would typically call fish are over here, but of course they share a distant common ancestor in the past with tetrapods that we obviously wouldn't consider fish. And so in that sense, you could call them fish because they had an ancestor that itself was a fish. But to me, it kind of, I think a better way to describe it would be based on our traits. And that means that we would have things like actinopterygians. I'm probably mispronouncing that, sorry. Basically the ray fin fishes. And that's what we typically considered fish, along with the cartilaginous fish. And the sarcopterygians would be including tetrapods and things like coelacanths and lungfish and things like that. And I think that's maybe a better way to describe it than saying we are fish or we had an ancestor that was fish, because at some point it gets kind of arbitrary. That's what I would say.
Okay, I appreciate that response, Andrew. Did you want a response as well, Neff? Anything you wanted to add to that question?
I would just say that sarcopterygian fish is a problem for evolution. The idea of fish evolution from to land-dwelling creatures doesn't support evolution theory. It's just full of gap, and we don't have a fossil record that demonstrates any such thing. From lobe fins to legs, and it's a leap. You have fish, then suddenly you have creatures that walk on land. There isn't anything in between.
I appreciate that, Neff. And the question was for you, Andrew, as we always do. You get the last word.
Yeah, same talking points about the fossil record. So I think I'm just going to leave it at that so we can get through as many questions as possible.
Okay, awesome. Next question here comes in from, let's see, Kevin Blah. Question, I assume, for you, Andrew.
Probably for me.
What is the, you're in the hot seat. I appreciate you being a good sport. Question from Kevin. What is the strongest evidence for macroevolution?
Well, that would probably be obvious. Now, some people out there would say there is no strongest evidence for macroevolution or common descent, which in a sense is true because you don't really have strongest evidence. You have a consilience of independent pieces of evidence. And it, to me, of course, what I would consider the strongest out of those would again be this nested hierarchy and the fact that we have statistical tests that have put common ancestry and a testable version of separate ancestry or common design against each other. And that's why I point out that extremely small p-value earlier. So the fact that we have a nested hierarchy, we know how it can be produced, and we know that separate ancestry, the simplest version of separate ancestry, doesn't produce that, would be the best piece of evidence for me.
Awesome. And Neff, if you had a response as well, go ahead. Neff, I think you're on mute.
Okay, I'm so sorry. I had to do something real fast.
No
Was I
worries.
asked a question? I'm sorry, I missed it.
Well, that's okay. You know what? We got so many questions here. Why don't we just go
Yeah.
right to the next one? No worries, no worries. And you guys don't have to feel obligated to always have a response, but just so the opportunity is there, of course. And we'll just always make sure that whoever the question is for gets the last word. So that being said, we've got a question here from Redefine Living. I appreciate the question. Redefine Living. Question 4, once again, I assume it's for you, Andrew. The question is, how does DNA barcoding affect the phylogenetic tree?
I'm like a black hole tonight. I'm sucking up all the questions. I from what I've heard about DNA barcoding, to me, it's based on some aspects of phylogenetic methodology. And in order to say phylogenetic methodology being used to discredit phylogenetic methodology seems kind of silly to me. That's really all I could say at this point. I don't know much more beyond that. Sorry, Redefine Living.
I appreciate it. I appreciate the question and the response. Neff, any thoughts on that specific question? I think you're on mute again, Neff. Neff, are you there? Here, let's go. He may have stepped away for a second. So let's just go right to.
I'm sorry, my cat is driving me bats.
Those
No
darn
problem,
cats.
no problem. It's all that cat phylogeny talk during the discussion, Neff. So.
If I missed a question, I'd like to answer it. Was there a question asked to me?
No. This specific question was to Andrew about DNA.
Okay, good. All right.
So let's go right to this question from Doki Doki SFT Fan Club. I appreciate the question and the support, Doki. So, question for Andrew: is mostly common ancestry or some common ancestry a compromise you would ever consider?
That's
so maybe. So I'll read it word for word. It's, is mostly common ancestry or some common ancestry a compromise you would ever consider? In other words, what I think Doki's asking is if just, let's say most organisms were related, is that, but not all, is that something you would consider? I think that's what the question's getting at.
I think I would respond to that by saying, to me, the only place you can really consider the most controversial would again be at the root of the free domains of life. And it's possible, conceivable to me at least, that maybe there are independent origins there. Now, of course, probably most phylogeneticists would disagree with that. So I feel like mostly common ancestry would be the only way I could settle it. I don't think that things such as phyla, or I mean, at least the phyla level, going back that far, like you can tell that things within a specific phylum of animals, like Arthropoda, for example, are all related to each other. At least the analyses don't disagree on that. So I guess so. It kind of depends. It really depends on how far you want to take the incongruencies and how far you really want to investigate them. If that makes sense. It just depends.
Yes, I appreciate that question, Doki Doki, and the response, Andrew Neff. If you wanted a response, go ahead.
Yeah, I'd say, when it comes to Arthropoda, the differences between the anatomies in various arthropods is profound. To believe that evolution could produce that is just philosophy. For example, some of the creatures that lived in the Cambrian the period have outrageously different anatomies, and they are in all manner of body plans. To evolution, this is a big problem because those creatures apparently popped onto the scene in no less than 500 million years, which is a drop in the bucket. Evolution has to operate on steroids to produce that. There's not even a mechanism for that.
Could I respond to that, SFT?
Yeah, and the question was for you, Andrew, so you can have the last word.
Yeah. So I would agree, Neff, that is a problem that needs to be solved. I think there is work being done on it, and to me, the GOBE, Great Ordovician Biodiversification Event, doesn't get enough attention nowadays. I think that does kind of contradict the claim that all the major animal phyla appeared then, because you have all these weird phyla that are kind of evolutionary experiments in a sense, if that makes sense. So that's what I would say on that.
All right, thank you from the both of you. Godzilla Freak is up with
good question.
the next question. So we've got it up on screen. Godzilla Freak, question for Andrew. Again, I appreciate you being a good sport. So far, all the questions are
Hey,
for you.
I'm the outlier here. I'm soaking up all the questions.
You're doing well. You're doing well. So the question for you, Andrew, is how do phylogenetic reconstructions decide where they root the trees?
Right. So as far as I'm aware, there are two basic methods to this. The kind of most obvious, but to me the most problematic, would be just looking back in the fossil record and seeing, when does this ancestral character state appear? If it appeared then and appeared before this thing, then that means that's the ancestral state, and that's where you root the tree, because you can say, this is the ancestral state, everything changed from this is the derived state, this is how we're going to root the tree. To me, I guess that could work, but to me the better method would be outgroup comparison. Like, you got your in-group, your group you're studying, like say for instance you have birds and dinosaurs, and they share feathers. And crocodiles, for example, the closest living relatives to birds, don't share that trait. They don't have feathers, obviously. So crocodiles in this case would be the outgroup. And since they don't have feathers, that would mean the ancestral state is no feathers, and which means the derived state is feathers, or has feathers. And that means that's how you root your tree, by having an outgroup that you know from other evidence is closely related to your in-group, and that's how you find the ancestral state and do it from there.
All right, great question, Godzilla Freak, and I appreciate the response. Neff, did you want to respond at all, or did you want me to go on to the next question?
Yeah, sure. The idea of evolution producing a feather is a no-go. It's just absurd to believe that random mutation is going to design something as functional and intricate as a feather. The evolutionists just make assumptions left and right. In the fossil record, it clearly does not support evolution. Countless prominent evolutionists, university professors, and paleontologists all have acknowledged in their own books that the fossil record doesn't show clear evidence of evolution. You have to imagine what you want to see when you look at it to believe evolution.
All right, I appreciate that, Neff. Did you want the final word on that one, Andrew?
I think I'm good.
Okay, well, let's go to this next question. It's kind of for you, Neff, but it's directed at you, though, Andrew, but you'll see what I mean when I read it. Okay. So the question is from Creo Debunk. Question for Rational Empiricist. You should ask why bat wing is more similar to mammal hand. What is similar to whale fin compared to bird and Archaeopteryx also are similar to these phyla? I hope that was worded okay. That is
I feel like I understand what he's saying.
Okay, did you want to reword that? It's kind of a question for you, though, Neff, so I'm not sure if...
Yeah, I'll be happy to answer it, unless he doesn't. Okay. Well, the idea, again, it just comes down to patternicity. Evolutionists see what they want to, and they assume relatedness. The idea, imagine the evolution of the wings of a bat. Evolutionists believe that a rat-like creature evolved into bats. Now, for this to have happened, the front legs of this creature, and through generations upon generations, had to get long and spindly and not very good for moving around on the ground. While at the same time, their rear legs got short and stubby. And this allegedly produced a creature that was viable, and natural selection would select it before it had the ability to fly. It couldn't fly yet, neither could it run properly, and a carnivore the size of a chihuahua comes along and chomp, you're out because you can't run anymore and you can't fly yet. So the idea of the evolution of bats or birds is just an absurdity. It doesn't happen incrementally. It doesn't even make sense that it could happen. And a creature that's not yet a bird, but not what it was anymore, would be a viable species that could survive, and natural selection would promote it.
I appreciate that response, Neff. Andrew, did you have any points or anything you wanted to add to that?
Yeah, I would just say that intermediate stages aren't necessarily half wing or half leg or things like that. They're more in terms of the form of the traits, not necessarily how complete they are. If complete even has a meaning in this sense, like you could say like here's I can't think of a good example. I'll just leave it at that.
Okay, I appreciate that. So let us end with this question, if I can find it up in the chat. Question for you, Andrew, from Alyosha. I appreciate the question. Why can't the evolutionist side be happy with evolution happening at the genus/species level? What are your thoughts on that, Andrew?
So the problem with evolution just happening at the genus species level, and this kind of gets into mechanisms of evolution versus common descent again, because sure, common descent is linked to some kind of evolution, not necessarily a specific mechanism. Is probably what I meant to say earlier. Basically, if you know that these species are related to each other again through phylogenetics, to something to things that are above the genus or species level, and you know that they're related by common descent, you can say some kind of evolution happened, that they had to change somehow. Now, what mechanism caused them to change, how they specifically, what environments they specifically occupied and started to migrate into, things like that, that's not necessarily related to that. So I don't know if that helps, but basically if they're related through common descent, then some kind of evolution has to happen above that level.
All right, I appreciate that answer, Andrew. Neff, any thoughts at all on the question or anything that Andrew may have responded with?
I'll just respond with it's all philosophy. We believe this is what happened because we believe that's what happened, and that's why we believe what happened. And oh, look, there's a genetic similarity. It's very distant between this creature and that one. That means they're related. We don't have to map a mechanism for it. We don't have to know what can cause it. We can point to things that cause disease and say that's a mechanism for it. But it's true. Trust me.
And as we always do, to be fair, Andrew, question was for you. If you'd like to have the last word, go ahead.
Yeah, I would just say the main assumption of phylogenetics in general, like if you go into the computer algorithms, they don't necessarily assume common descent. They're just looking for a tree in general. But phylogenetics overall, it does in a sense assume common descent. The problem is that the assumption is warranted again because we have these nested hierarchies and we know the thing that can produce them is common descent. So they're related and linked, but it isn't circular. It's kind of, if this is the case, then this happens. We see this happening, so this is the case because that's the only known thing that can produce that pattern.
All right. Well, I appreciate it. Great job to the both of you answering these questions. Very important questions. Very important debate. Tons of fun. It's already been 2.5 hours, nearly
Wow.
2.5 hours. So, as always, these debates fly by. Lots of good points and topics discussed. To the audience, a couple reminders before we shut it down. We'll be back here on Monday with Dr. Joseph Kazil. He is going to be giving a presentation titled "You've Come a Long Way, Baby." We are also going to have an audience Q&A for that. So make sure you are there and make sure you are bringing your questions. Also, the middle of the month, another reminder, the big debate, the age of the earth, Rhodes versus Hovind, and the day before that on the 14th. So we're going to have a couple awesome days in a row. Dr. Frank Turek will be here. We will be discussing his book, I Don't Have Enough Faith to Be an Atheist. So that being said, another great debate in the books. Guys, please, if you love debates and you are a debate addict like all of us on the panel, go to the playlist section titled Debates Hosted by Standing for Truth. We are now over 100 debates, 100 plus debates, all sorts of topics, theology, science. So check that out. It'll keep you busy for the rest of the year. That being said, gentlemen, thank you again for giving us your time for tonight's debate. I want to hand it to you guys, Andrew and Neff, for some final words, final thoughts before we shut it down. Why don't we start with Neff? Once again, thanks for being here. Thanks for doing this.
Oh, thanks for hosting it, brother. I'm going to have an after show, I guess. I'll fire one up if anybody wants to come to my channel, and you can join or listen. I prefer you join. My final words is, I guess, that evolutionists are welcome to believe what they want about genetic similarities between different creatures, but unless they can show a mechanism that means that genetic similarity is produced by evolutionary processes and not evidence of common design, they have nothing. That's all I have to say.
Awesome. Well, once again, thanks for doing this, Neff. Thanks for your final words. Rational Empiricist, to you as well. Thanks for giving us your time for tonight's debate. Thanks for doing it. These discussions and debates are important. So just because we disagree on certain topics, we like to kind of get into the octagon, as we could call it, to battle out these ideas and discuss these topics. So Andrew, thanks again. And any final words from you?
Yeah, I would just say debates not really my thing, because I guess whether or not me and Neff disagree on this topic, reality is reality, and the scientific community, who knows, maybe someday it'll find a common design pattern, but that's just not what the evidence tells us today. And until the evidence tells us that and the scientific community starts hopping on that road, I don't know, it seems kind of fishy to me. So
all right, well,
stay curious, as some would say.
Awesome. Well, thank you. Thanks to the both of you for a debate to remember. It looks like NephilimFree is having an after show. So Doki Doki has put that link in the description box. So anybody interested, please go over to Neff's channel for the debate after show. That being said, we'll see you on Monday. And God bless everybody in the chat. Thank you for all your support, super stickers, super chats, and questions. SFT is out.